In metaphase cells, the microtubules of mitotic and meiotic spindles are highly dynamic, yet the spindles as a whole maintain a consistent length. This is partly due to a balance in the forces produced by microtubule motor proteins. Some motors push the spindle microtubules out towards the poles, a process known as microtubule flux. Other motors counteract this force by pulling the poles back inwards. But a collection of non-microtubule, non-motor proteins called the spindle matrix could also contribute to the mechanical forces within metaphase spindles. Jay Gatlin of the University of North Carolina explains more.
What is the spindle matrix? I think many of us have different ideas. I think that in order to be defined as a spindle matrix component, you have to have dynamics that are distinct from tubulin or microtubules. You have to be somewhat autonomous of microtubules. They don't require microtubules to be there. And oftentimes when you get rid of these components, the spindle either falls apart or you have problems with chromosome congression to the metaphase plate and other issues, which suggests they're definitely playing a role in spindle assembly. But the other part is whether or not these components provide some sort of mechanical contribution to spindle shape, specifically spindle length. And that has been something that's been very difficult to test.
Gatlin, a postdoc in Ted Salmon's laboratory, directly probed the mechanical properties of the spindle matrix by skewering spindles in Xenopus egg extracts with microneedles.
Our rationale was that if we stuck a needle into this poleward-flowing microtubule lattice, presumably with a lot of different molecules that are cross-linking the spindle microtubules, that a needle would get caught up in that flow and get pushed poleward. Or because the needle can't move in this case, the spindle actually pushes itself off the needle. And if there is a matrix there, we're also pushing that same skewering needle right through the matrix. So if there was this really static and strong matrix, our prediction was that maybe nothing would happen, that flux would continue unabated and the spindle wouldn't move. And then the other prediction was that perhaps the spindle would just push itself right off the needle. You would simply see the needle flow off toward the pole at a flux-like speed, and that would indicate to us that there was little resistance provided by a matrix.
So how much mechanical resistance would the spindle matrix offer as the poleward flux of microtubules pushed the needle towards the poles? The answer was not a lot.
What we found was that the spindles pushed themselves off the needle with rates that were well within the range of reported values for flux, somewhere on the order of 1.7 microns per minute.
In other words, the spindle matrix provided little resistance to the poleward flow of microtubules, at least in the middle of the spindle. More resistance was offered as the needle neared the spindle poles, however.
As the needle got closer to the pole, the rate slowed down appreciably. So it went down to 0.8 microns per minute. So we thought maybe there's some different architecture at the poles that was causing more resistance there, slowing the rate at which a needle could move through it. But the other problem in the literature is that flux slows down at the poles as well. So we thought, okay, is what we're seeing the result of our pushing mechanism slowing down at the poles, or is it the result of some sort of different kind of structure there, some more robust structure at the poles?
To find out, Gatlin and colleagues inserted needles on either side of the metaphase plate and dragged them rapidly outwards to stretch the spindles.
And what we found was that both needles would flow right through the spindle without any sort of obvious deformation of the spindle until the needles reached the poles. And at that point, it was obvious that they had sort of gained purchase on something, and we could start stretching the spindles.
This suggested that there was something different about the mechanical properties of the poles, a conclusion that was also supported by experiments in which needles were pulled apart along the axis of the spindle equator. The outermost microtubules remained attached at the poles and dragged them inwards.
Again, it supported the idea that there's something structurally distinct about the poles, structurally sort of more robust than anywhere else in the spindle. But in the context of spindle length regulation, it's sort of difficult to envision how a matrix that's confined to either end of the spindle might be contributing to spindle length.
Whatever this spindle pole matrix is doing, it's likely that it consists of the protein pneuma, which is known to build an insoluble matrix-like structure at the poles in conjunction with the motor protein dynein. When the researchers repeated their skewering experiments in the presence of an antibody that inhibited dynein function, the spindle poles lost their structure, and the rate of spindle movement no longer slowed as the needle reached the spindle ends. So there may be a reasonably strong matrix structure at the poles, but it seems the spindle matrix as a whole doesn't make a significant mechanical contribution to spindle length, unless, says Gatlin, it's organised in an unexpected way.
When you read about the matrix, or you sort of picture a matrix in your own mind, implicit in the definition is that it's sort of this meshwork-like thing that runs in and out of the spindle, and the properties of this thing are invariant as far as the orientation. What our data suggests is that at least along the interpolar axis, there's little resistance to needle movement. Our experiments do not rule out the possibility that there's a matrix that aligns itself with the microtubules. It's difficult to tell whether or not that's the case. So what our data really does is it provides limitations on how a matrix can look. So it really suggests that if there is a matrix that's regulating spindle length, it has to be anisotropic. It has to align itself structurally with the microtubules, or at least be very strong in that direction and very weak in the other.
It remains to be seen if any matrix components are organised in this way. In the meantime, you can read more about the mechanical properties of the spindle matrix in the paper by Gatlin et al. In the February 22nd issue of the Journal of Cell Biology.